Hello students, welcome to EPG Pathasala. I am Dr. P. Nagaraj, Assistant Professor from Anna University, Chennai. Today we are going to talk about module on protein-DNA interactions from the paper Structure and Function of Biomolecules. Today lecture objective is basics about proteins and its structure, introduction of proteins and DNA, basic function of proteins, function of DNA. There are specific interaction and non-specific interactions, also importance of those interactions that we will study in details. All living things are made up of four classes of large biological molecules. Number one, carbohydrates, and second one is lipids, third one is proteins, and fourth one nucleic acids. Macromolecules are large molecules composed of thousands of covalently bonded atoms. Molecular structure and functions are inseparable. That means the structure and function both are complex in nature. Proteins includes a diversity of structures resulting in a wide range of functions. Proteins account for more than 50% of the dry mass of the most cells. Protein functions includes structural support, storage, transport, cellular, communications, movements, and defense against foreign substances. These are all the functions of proteins. Enzymatic proteins. Function of enzymatic protein is selective acceleration of chemical reactions. Examples are digestive enzymes, catalyzes the hydrolysis of bonds in food molecules. That means enzyme bind to the activation place that will be released after the reaction is over. Storage proteins. The function is storage of amino acids. Example, caseins, the protein of milk, is the major source of amino acids for baby mammals. Plants have storage proteins in their seeds. Ovalbumin is the proteins of egg white used as an amino acid source for the developing embryo. Hormonal proteins, that function is coordination of organisms' activities. Example, insulin is just a hormone which is also a protein also. A hormone secreted in the pancreas causes other tissues to take up glucose, thus regulating blood sugar concentrations. So this insulin is responsible for regulating blood glucose levels. Then defensive proteins, another class of proteins. The function of protein is protection against disease. That is the main role. Example of defensive proteins are antibodies, inactivated and help destroy viruses and bacteria. The antibody role is it will make a binding between antigens, that means virus or bacteria, then it will destroy the entire organism via antibody antigen interactions. Transport proteins, this is another role of proteins where the main role is to transport of substances. Examples are hemoglobins, which is again protein, the iron containing protein of vertebrates. Blood transports oxygen from the lungs to the other part of the body. Other protein transport molecules across the cell membranes. So the very important phenomena is transport substance or molecules from one place to entire body. That is called transport proteins. Receptor proteins, that means the response of cell to chemical stimuli. It will receive the signals as well as process. Example, receptor built into the membrane of nerve cell detect signaling molecules released by other nerve cells. That means the signaling molecules will give nerve cells as a receptor protein. This is a function of another class of proteins. Structural proteins, example which will give more support to the protein molecules. Function is to give the support. So well-known examples is a keratin, is the protein of hair, horns, feathers, and other skin appendages. Insect and spiders use silk fibers to make their cocoons and webs respectively. Collagen and elastin proteins provide a fibrous framework animals' connective tissues. So this is structural, again will give a structural stability or support to the protein molecules, which is known as structural proteins. Enzymes are a type of protein that act as a catalyst to speed up chemical reactions. So enzymes are similar to catalyst. In case of chemical reactions, catalyst will activate the reactions. Similarly, in case of biochemical reaction, which is activated by enzymes, which will never involve or participate, it will speed up the reactions. Enzymes can perform their functions repeatedly, functioning as workhorse that carry out the process of life. So entire our body biochemical reaction happen because of enzyme present in our body cells. Amino acids are organic molecules with the carboxyl and amino groups. That is very important in the same carbon probably. Amino acids differ their properties due to differing side chain called R groups. That means amino acids, one carbon having amino group as well as carboxyl group. If both are present on the same carbon, which is called alpha amino acids. Whereas if the acid group amino group separated by one or two carbon, which is known as beta amino acid, gamma amino acid, depend upon the linkage or attachment of amino group, acid group between the carbons. Apart from that, the side chain which will play major role in case of amino acids. Polypeptides, peptides which is formed by amino acids combined together will give a peptides. In case of polypeptides are unbranched polymers built from the same set of 20 amino acids. It is very important. A protein is a biologically functional molecule that consists of one or more polypeptides. Polypeptides is nothing but the molecules will combine together to form peptides. The peptide molecule will combine together will give polypeptides with the elimination of water molecules. Nonpolar side chains. The amino acids having some of the nonpolar side chains like hydrogen, methyl and isopropyl and butyl, something like that, which is again nonpolar in nature. Aromatic group, thiol group, indole, etc. will give nonpolar side chain, which means that solubility of the all amino acids which will be less favorable in water or more favorable in organic solvents. Polar side chains also possible in case of amino acids like serine, threonine, cysteines and tyrosines, asparagines and glutamines. These are all examples for polar side chains. That means which is hydrophilic in nature, which will more towards water soluble rather than organic solvent soluble. Electrically charged side chains also known as hydrophilic. That means some of the amino acids having acidic or negatively charged groups, some of them having basic or positively charged groups. Acidic amino acids are aspartic acids or glutamic acids, both are having acidic functional group, whereas in case of basic functional groups are lysines, arginines, histidines having a basic unit of amino acids group. Amino acids are linked by peptide bonds. That means two amino acid will link via peptide, then peptide will link it via again peptide bond will produce polypeptide. Polypeptide which will give the proteins. A polypeptide is a polymer of amino acids, and that is what we discussed. And polypeptide range in length from a few more than 1000 monomers. Each polypeptide has a unique linear sequence of amino acids with a carboxyl end C terminus and an amino end or N terminus, which is discussed in the details. The peptide bond, that means acids of one amino acid group and amines of other amino acids group with elimination of water or dehydration reactions will give NHCO bond, that is generally known as peptide bond. That is one acids and one amino group combinedly together will produce peptide bond. There are two peptide bonds which again two molecules which will combine together with another amino acid with the elimination of water will produce tripeptide. Again will make one more peptide bond, will give 3 or 4 amino acid. Again it will combine with other amino acid will produce tetrapeptide. Like this the peptide chain will extend based on that whatever amino acid present it will produce a peptide. If more number of peptide will combine together will give polypeptide. At first all we have is a string of amino acids bound with peptide bonds. Once the string of amino acids interact with itself and its environment. Then we have functional proteins that consist of one or more polypeptides precisely twisted, folded and coiled into unique shape. The sequence of amino acids determines a protein three-dimensional structure. A protein structures determines its functions. So first the structure of protein will be determined by the sequence of arrangement of amino acid. That arrangement amino acids will give the function of proteins. Especially primary structure which is consist of its unique sequence of arrangement of amino acids, and in case of secondary structure, of course, found in most proteins consist of coils and fold in the polypeptide chain, that is like alpha and beta. And tertiary structure of protein is determined by interactions among various side chain, that whatever side chain present in the amino acid that have interaction, that will make a entire tertiary structure. Whereas in case of quaternary structure results when a protein consists of multiple polypeptide chain, that means more than two chains, polypeptide chains interacts together will give a bulk structural change, that is called quaternary structure. Primary structure of proteins, the sequence of amino acids in the protein is like the order of letters in long word. Primary structure is determined by inherited genetic information. The primary structure basically will give sequence of arrangement of amino acids, how it is arranged in the protein molecule. That will give, we can get information based on primary structure of proteins, which is gives a secondary structure of proteins. The coils and folds of secondary structure result from hydrogen bonds between the repeating constituents of the polypeptide backbone. Why the secondary structure arising in amino acids or protein molecule? Because of based on hydrogen bonding. Typical secondary structures are a coil called an alpha helix and a folded structure called beta pleated sheets. Two forms of secondary structure available in case of proteins. Secondary structure also two ways. One is alpha helix, that means the helical structure will give alpha helix, coiled structure of amino acid because of presence of hydrogen bonding. In case of beta pleated sheets, the layers of helicals which will combine together will give a sheet like a structure, that is in beta 10, which is known as beta pleated sheets, again which is also based on hydrogen bonding between two layers of protein layers. In case of tertiary structure is determined by interaction between the side chain present in the amino acid groups rather than interaction between backbone constituents. This interaction between R groups includes actual ionic bonds and strong covalent bonds called disulfide bridge, which may reinforce the protein structure. So we already discussed where amino acids having sulfur thiol, which will make two link between disulfide bond, that will also play in the major role in case of tertiary structure. Apart from that, IMFs such as London dispersion force, van der Waals interactions, hydrogen bonds and hydrophobic interactions may affect the protein structure. Based on deals with the tertiary structure of proteins, again the hydrogen bond present in the carbonyl group of one molecule and hydroxy group of other molecules, and similarly amino group of nitrogen, and similarly disulfide bridge. These things will give tertiary structure of proteins, which is hydrophobic interactions and van der Waals interactions, main reason for getting tertiary structure of proteins. Quaternary structure results when two or more polypeptide chains form one macromolecules. Collagen is the fibrous protein consists of three polypeptide coiled like a rope structure, which is examples for quaternary structure of proteins. Hemoglobin is the globular protein consists of four polypeptides, two alpha and two beta, which is represented both combinedly four together will give hemoglobin. Will give primary structure, which is sequence of arrangement of amino acids. In case of secondary structure will give alpha helix and beta pleated sheets. In case of tertiary structure will understand the domain. Quaternary structure will give overall the entire protein molecules. A slight change in primary structure can affect a protein structure and ability to function. That means small change in any one of the amino acid will not work in that particular function. Example, sickle cell disease, an inherited blood disorder, forms a single amino acid substitution in the protein hemoglobins. That means you change small part of amino acid, then entire function and properties may change and it won't do the same function what earlier it did. In addition to primary structure, physical and chemical conditions can affect the structure. Alteration in the pH, salt concentrations, temperatures or other environmental factors can cause a protein or unravel. This loss of protein's native structure is called denaturations. A denatured protein is biologically inactive. That is the problem. The proteins are enzyme which is substrate specific, temperature specific and pH specific. If you change any one of the condition, that protein will undergo denaturations. Once the protein undergo denaturation, that may be in no more active, that is called inactive form of proteins. In case of nucleic acids, again which is stores, transmit and help express heredity informations, and that is the genetic informations. And of course the amino acid sequence of polypeptide is programmed by a unit of inheritance called a gene, and genes are made of DNA. A nucleic acid made of monomers called nucleotide. Nucleotide is a monomer which will make a polymer chain, will produce a polymer, that is called nucleic acids. There are two types of nucleic acids. One is deoxyribonucleic acids and another one is ribonucleic acids. In case ribose, the C2C3 having the hydroxy group, whereas deoxyribose, the C2 portion doesn't have a hydroxy group. Only that is the difference between these two molecules. DNA provides direction for its own replications. DNA direct synthesis of messenger RNA, mRNA, and through mRNA controls protein synthesis. Protein synthesis occurs on ribosomes. So the basic difference between ribose and deoxyribose is the second carbon having the hydroxy group. If it is present, that is ribose. If not present, that is deoxyribose. Each nucleic acid is made of monomers called nucleotide. Nucleotide means that is phosphate unit, sugar portion and nitrogen base. All together will combinedly called nucleotides. Each nucleotide consists of a nitrogenous base, pentose sugar and one or more phosphate groups, which will give the simplest example of nucleotides, where the sugar portion and nitrogen base and phosphate combinedly together some monomer which will link it together with the other monomer unit will produce a polymer chain. The nitrogen bases are based on two types. One is pyrimidine and another one is purines. Pyrimidine bases are 6-membered nitrogen heterocycles. Purine bases are indole type fused rings, which is adenine, guanine are purine bases. Cytosine, thymine, uracils are pyrimidine bases. Double strand DNA is two ways based on hydrogen bonding. Either it may be parallel or it may be anti-parallel, both way it is possible. So which is again the phosphate units linked together with other unit to make the polymer chain. There are two families of nitrogen bases, pyridines and purines, which we have already discussed. In DNA the sugar is deoxyribose, in RNA the sugar is ribose. Adjacent nucleotide are joined by covalent bond that form between the OH group of 3' carbon of one nucleotide and the phosphate on the 5' carbon on the next. These links create backbone of sugar phosphate units with nitrogen bases as an appendage. The sequence of base along DNA and mRNA polymer is unique in each genes. RNA molecule usually exists as a single polypeptide chain. DNA molecules have two polynucleotides spiraling around an imaginary axis forming a double helix. In the DNA double helix, the two backbones run in opposite 5' to 3' directions from each other. An arrangement referred to antiparallel. One DNA molecule includes many genes, 5' to 3', 3' to 5', which is antiparallel to each other. The nitrogen bases DNA pair up and form hydrogen bonds. Adenine always make hydrogen bond with thymine. in case of guanine always make with cytosines called complementary base pair. Complementary pair can also occur between two RNA molecules or between part of same molecules. RNA thymine is replaced by uracils in case of RNA. This represents the RNAs, transfer RNA and base pair joined by hydrogen bonding. The linear sequence of nucleotides in DNA molecules are passed from parents to offsprings. Two closely related species are more similar in DNA than are more distantly related species. Molecular biology can be used to assess evolutionary kinships. Possible forces used here: define the persistence length of nucleic acids, know 4 forces used in protein nucleic acids, interactions and the details of each, know how DNA shape affect its ability to be bound by proteins, know other geometric concentrations of double-strand DNA, know geometric constraints of single-strand nucleic acids. Understand how proteins interact non-specifically with the DNA and RNA, achieve proteins, and gene 5 from page, and RNase A some slight specificity, and DNase 1 polymerases E. coli, which is HIV RT. Protein nucleic acid interactions, again forces are electrostatic forces, dipolar, that means hydrogen bonds, hydrophobic and dispersion force. In case of electrostatics, salt bridge, 40 kilojoule per mole of stabilization per salt bridge. In case of negative phosphates, DNA RNA with positive amino acids, lysines, arginines and histidines, which is responsible for this kind of force. Influenced by salt or salt bridge, the pattern of salt bridge can distinguish between single strand and double strand DNA RNA, and between bDNA and zDNA also can be distinguished via salt bridge. The overall electrostatic field of proteins can orient polyanionic nucleic acids. Modeling of interactions can also be identified. Protein nucleic acid interactions. Forces, what are the forces it can be used? It may be electrostatic force, dipolar or hydrophilic, hydrophobic, dispersants, etc. Dipolar phase, between two polar charges are separated. Hydrophobic, because of the attachment of the side chain, that will make hydrophobic. In case of hydrophobic interaction, dispersants also occurs. Protein nucleic acid interaction against the geometric constraints, double strand DNA, high charge protein domains that interact with it have a complementary surface, polar charged chains interact with the phosphate oxygens. So this is how the interactions: polar side it will interact with the polar part, non-polar side it will interact with non-polar part. Geometric constraints between double strand bDNA. Alpha helix interacts in major groove with base, most common because of hydrogen bond donors and acceptors in major groove. Interaction involved that can be identified using protein nucleic acid interactions. Again, the interactions of geometric constraints, single strand nucleic acids, hydrophobic base exposed will have more phobic nucleic acid binding surface. That means that the geometry itself it will give whether it will be hydrophilic or hydrophobic. If it is hydrophilic, which will interact with hydrophilic sites. If it is hydrophobic, that will interact with hydrophobic interactions. RNA, lots of regions of single strandness A form major groove, deeper and base more inaccessible. Bulges can help with major groove. Again, this will give lots of regions of single strands, bulges, loops, doconates. Loop structures can bind protein together. Which again non-specific interactions, protein nucleic acid a specific one and this is a non-specific. Exonuclease and endonuclease RNase, a cleaves RNA but also binds single strand DNA, competitive inhibitor, group of positive charged residues on proteins. Electrostatics are main force involved, so little sequence specific for sequence with 5 days to cleavage its since thrombin 45 H bonds. That means that hydrogen bonding will be the major issue of making this kind of protein nucleic acid interactions. Again, non-specific interactions, exonucleus, endonucleus, DNase forces, little sequence specific, structure of complex exposed loop of enzymes binds at minor growth of DNA which mimics naked DNA. Protein nucleic acid interactions of non-specific interactions, DNase forces contact of hydrogen bond salt bridge, little sequence specific. Deals with non-specific interactions, the DNA dependent DNA polymerases, example E. coli DNA, pole 1 and 3, pole 3 chief replicative of beta subunit of pole 1, repairs damages DNA and converts Okazaki fragments into complete genomic DNA, large fragments, small subunit. Also deals with the protein nucleic acid interactions, which is again comes under non-specific interactions, DNA dependent DNA polymerase and position of OH attack phosphorus and generate pentacovalent transition state, stabilizes leaving groups. Also deals with protein nucleic acid interaction of non-specific interactions, RNA dependent DNA polymerases, uses DNA or RNA templates 3 days to 5 days proofreading, and that will also be kind of interaction. Discuss also protein DNA interaction of importance. We can identify the gene expressions by transcription initiation, tata binding protein, RNA synthesis, RNA polymerase, transcription regulations, MAX proteins, DNA repair or DNA glycosylase, oxidative DNA damage. These are all the information we can identify using protein DNA interactions. Predicting DNA binding function. Knowing a protein function is essential in understanding cellular locations, interactions, biochemical pathway, potential as drug targets. Prediction of protein DNA binding site given unbound protein structure. This is again electrostatic patches or motifs. Predicting functional form of structures. Structural genomics, filling in the gaps of protein structure phase. Structures solved that they have low sequential identity, that means less than 30% sequence identity, potentially little or no fold similarity to any currently in the PDB, require algorithms to make fast and reliable functional predictions. So students, let us now summarize what we have learned in these models. So protein-protein interactions, a method of combined structural data from NDB and PDB with the sequence data from PFAM and SMART. A structural template library of 7 HDH motifs and recognition across the families, and a template method in dependent and global fold similarities, potential to identify new DNA binding HDH motifs. All these things etc. we studied in our protein DNA interactions.